Thank you for joining us for this MDA Community Education webinar. My name is Michelle Barrios, and I'm the Community Education Specialist at MDA. We are thrilled to have you join us today for this important and educational on-demand webinar. This webinar today is part of our larger MDA Engage flagship community event series, which focuses on bringing the neuromuscular disease community together around education and social opportunities. Be sure to visit the MDA Engage section on mda.org for updates on upcoming virtual education events. Before we begin, I would like to say thank you to our speakers, whom you will meet shortly. Also, thank you to our event supporters, Genentech and Mitsubishi Tanabe Pharma America. We would not be able to provide events like this if not for their generous support, so thank you very much. The Muscular Dystrophy Association is committed to transforming the lives of people affected by muscular dystrophy, ALS, and related neuromuscular diseases. We do this through innovations in care and innovations in science. MDA's research is vast. We are aggressively working to advance therapy development through support of innovative research in universities and industry and collection of healthcare data in the Mover Data Hub. MDA is the largest non-governmental funder of neuromuscular research in the country. A single breakthrough can lead to a cure, and in 2019, MDA supported 252 research projects worldwide. Our network model means findings from one disease often enable progress in others, maximizing the speed at which we can make progress. To share a little bit specifically to MDA's work in the clinical trial landscape for neuromuscular disease, in 2019, MDA awarded 25 new grants totaling more than $6.6 million towards research focus on a variety of neuromuscular diseases. Since its inception, MDA has invested more than $1 billion in research to uncover new treatments and cures for neuromuscular diseases. With that foundation, let us review the objectives for today's webinar. Participants will learn the process of taking a treatment from clinical trial to FDA approval, and listen to a firsthand clinical trial experience from a physician. Next, I would like to introduce our first two speakers. Dr. Hesterly is Executive Vice President, Chief Research Officer of the Muscular Dystrophy Association. She has over 20 years of experience in neuromuscular research in both the nonprofit and industry space. She has served as head of research for Parent Project Muscular Dystrophy, the Myotonic Dystrophy Foundation, the Association for Frontotemporal Degeneration, and the Muscular Dystrophy Association, MDA's venture philanthropy. Dr. Hesterly has also served as a project lead for rare disease gene therapy programs at Pfizer Inc., as Chief Executive Officer of Lion Therapeutics, a special purpose entity of AskBio Inc., and as Executive Vice President and Head of the Neuromuscular Division of AskBio Inc. She has been involved in numerous efforts to remove barriers to therapy development for rare disease and foster interactions between patient advocacy groups and industry. Dr. Hesterly has served on the governing board of the Health Research Alliance, the NINDS Council, and the Department of Health and Human Services Muscular Dystrophy Coordinating Committee. She currently serves on the congressionally directed medical research programs Duchenne Muscular Dystrophy Programmatic Review Panel. She received her PhD in neuroscience from the University of Arizona in 1999. Dr. Jerry Mandel is the founder of the Clinical Translational Gene Therapy Program at the Research Institute at Nationwide Children's Hospital, Professor of Neurology and Pediatrics at The Ohio State University, holds the Curran Peters Chair of Pediatric Research. He is a clinician scientist directing laboratory projects and carrying these to the bedside at the Abigail Wexner Research Institute at Nationwide Children's Hospital. He has published more than 375 articles with a focus on neuromuscular disease and authored books on disorders of muscle disease, nerve disease, and gene therapy for muscle disease. Dr. Mandel was the first to perform gene therapy for DMD, March 2006. He also initiated gene therapy studies in limb-girdle muscular dystrophy type 2D, showing sustained gene expression for more than 6 months, an important milestone for the field demonstrating potential for gene delivery with safety and efficacy. Dr. Mendel led the clinical trial on exon skipping, the first therapeutic agent to show increased dystrophin expression in DMD. And now, more than a decade later, dystrophin expression levels are still increasing, and follow-up long-term exon skipping outcomes demonstrate slowing in disease progression. He was the principal investigator on the published article on SMA gene therapy that showed unequivocal efficacy. This was a milestone achievement because it saves the lives of infants when the gene is delivered in AAV9 in very high dose by intravenous administration. In addition, an important finding was that pre-administration of prednisone 1 day prior to delivery with extension for 30 to 60 days significantly controls hepatotoxicity. Use of prednisolone in this way has now become the standard in gene therapy trials. This work in SMA received Science Magazine 2017 Breakthrough of the Year Achievement Award and the Clinical Research Forum Distinguished Clinical Research Award. Based on this study, newborn screening for SMA is now established in 31 states throughout the country. Currently, Dr. Mendel is actively engaged in systemic delivery of mycodystrophin in a DMD gene therapy and is the first to demonstrate efficacy in this wasting disorder, recently published in JAMA Neurology.
Thank you, Michelle. So today I'm going to talk to you a little bit about the role of regulatory agencies in clinical trials. If you participate in a clinical trial, you will certainly experience some of the terminology, and you will ideally have a better idea after this talk what the role of these regulatory agencies is in the different ways that they are involved in clinical trials. So just as a very busy slide, just to talk about the fact that drug development is a highly regulated process. It can be a long process. It can take as long as sometimes 20 years from the time that someone first has an idea that's in basic research all the way through a new drug approval. You can see that from the time you get to sort of proof of concept testing in animals through the new drug approval, the Food and Drug Administration is involved, and they're involved in a lot of different ways. Even before you get to a clinical trial, they still have standards for how you should gather and report data called good laboratory practice. Later, when you get to a clinical trial, they have standards around good clinical practice that you have to maintain. And as you get into clinical trials in the clinical testing space, there are other regulatory agencies that come into play as well, including institutional review boards and data safety monitoring boards. There are also specific things that sponsors of clinical trials have to file, including an investigational new drug application to test a drug that's for the first time in a disease indication, and a new drug application to get approval of that drug to be able to market and sell it. So we'll talk a little bit more about all of these regulations and how they come into play. So what the Food and Drug Administration does, more or less, is responsible for the safety and efficacy of human drugs, biological products, medical devices that are in our supply chain for the nation. So they interpret all the rules and laws that govern drug policy, and they also review data that's submitted by drug developers, ultimately to determine if the benefits of a new therapy outweigh the risk. So why do we need the Food and Drug Administration? So if you take a short look at the history of the FDA, and I'm certainly not going to read through all of these bullets, but just to say that in general, the history of regulation of drugs in this country has been fairly reactive. So in the early 1930s, there was a drug called sulfanilamide that was sold as an elixir legally, but they changed the formula of this drug and it suddenly started killing people. One doctor testified that it had killed 6 of his patients and one of his best friends. And it was at that point that Congress really enacted the 1938 Drug Act, which was designed to increase safety requirements for new drug testing. And again, later in 1962, a drug called thalidomide, which was designed to help with nausea during pregnancy, caused birth defects all over Europe, but not in the US, as it wasn't approved there. But it still prompted the FDA to strengthen the requirements further to prevent something like that from ever happening in the US. There were subsequent laws passed over time as well that strengthened the role of the Food and Drug Administration each time. Finally, in 1992, the Prescription Drug User Fee Act was passed by Congress to collect some funds back from the various companies and sponsors of new drugs. And those fees are designed to help the FDA operate. And that law actually contains a lot of information about how the FDA regulates drugs. And it's reauthorized periodically. And it often brings with it opportunities to change how the FDA does its job. So there are regulatory agencies all over the world, as you might imagine. Other countries have their own regulatory agencies. This is important, particularly in rare disease, because usually drugs aren't just approved in this country. They have to be approved in multiple countries, usually just so that there is a large enough number of people who could receive the drug for the company or the developer to be able to recoup the cost of making the drug. So all of these organizations have their own set of rules and laws that they follow. They're somewhat harmonized, and there are various alliances and efforts underway to try to harmonize those further so that to increase the efficiency of doing this, so you're not starting from scratch at each individual country. A good example of this is the EMA, the European Medical Association, which governs multiple countries in Europe under the same existing law and makes that a very efficient process. So for the most part, when you're testing a new drug, the risk and benefits are demonstrated through clinical testing. So clinical testing uses human subjects. So if you have been invited to participate in a clinical trial, this is where you come in, following this long period of testing in animals and gathering research and petri dishes. When the drug developer finally feels confident to test a drug in the clinic, the FDA has to approve that. The goal is to determine what is the benefit of the drug, whether it has side effects, how severe those are, and how to manage them, what dose of the drug you should give, and finally, how long the drug stays in the body and how it's broken down. So all of these things have to be determined in people before it's legal to market the drug. So just before we talk a little bit more about clinical trials and regulatory requirements, just a little bit of useful vocabulary. So you've heard me use the word sponsor. So a sponsor of a drug trial is really the entity that's legally responsible for that trial. And it's usually an institution or company. You will probably hear it most often in reference to a company as the drug sponsor, but it could be an individual or an institution. The clinical investigator, on the other hand, is the individual who actually conducts the investigation. This will be the person who gives the drug and is almost always a medical doctor. You sometimes might hear the word site PI or principal investigator of a site. So when you have a clinical trial with multiple locations or sites, there will be a principal investigator at each of those sites. An investigational drug is a drug being studied in a clinical trial. So a drug is not a drug. It's not a treatment. It's not a therapy if it's not approved. It's still an experiment. So it's an investigational drug or investigational product. Placebo is another thing that I'm sure you've heard. There's sort of the bane of the existence of anyone participating in a trial, but a placebo is a substance that's ideally not distinguishable from the drug product. And the goal there is to make sure that the drug benefit that you see is real and that it's not something that can be influenced by someone's positive ideas of the outcome. This is particularly important in neuromuscular disease, where a lot of the functional outcomes are effort-based. There are things like the 6-minute time walk or various other types of functional measurements, where really, if you are feeling confident, you might do better in a trial like that. It's not quite as objective as some other types of measurements. So placebo can be a very important way to determine that the drug is providing that benefit, not just well wishes. Biomarker is another term you might hear in the context of clinical trials. So this is something that you can use to give you an idea what an outcome is going to be like without, it's sort of a surrogate for that outcome. So for a good example of a biomarker, cholesterol levels, which have long been used as a way to understand risk factors for coronary or cardiac health. What you really want to know is whether or not you're going to have a heart attack. Your cholesterol levels may help predict that. So get you the answer faster than waiting for your actual outcome. And then finally, a clinical endpoint or outcome is something that you can measure that will represent a direct clinical benefit. The FDA requires that you have an outcome that's clinically meaningful. So something you can measure that the drug does that improves your life in a way that's meaningful to the person participating in the study. So just a little background there, because I will use some of these terms as we go forward. So quickly, the phases of clinical testing. You've probably heard phase 1, phase 2, phase 3. I won't go into them in detail here, except from the standpoint of where the FDA becomes involved. So before you can test a new drug, the sponsor of the drug must first submit an investigational new drug application. So this is a huge dossier that collects all of the data from the animal testing, including toxicology testing, all of the proof of concept testing. There's a lot of information about manufacturing of the drug in that application. That goes to the FDA, and they don't actually approve it. What happens is they have 30 days to decide if they're willing to let this trial go forward. So if you don't hear from the FDA within 30 days, you're good to go. On the other hand, they can look at the information you submitted and place the trial on hold. And frequently, they will have additional questions before they release it. They may even ask for additional data. So if you go forward, the phase 1 study is largely safety testing. Often it's done in healthy volunteers, but given the, with different types of therapy, depending on what it is, it might also, the phase 1 could take place in people who have the disease. Phase 2 or a phase 1/2, sometimes these phases you'll see slashes because they blur together. But a phase 2 is typically still primarily focused on safety, but it also starts to collect data, preliminary data on how well the drug works and what the dosing schedule is like. These phase 2 will typically test the drug in the people with the condition that it's meant to treat. And you can see also that phase 2 is typically bigger than a phase 1 study. And finally, a phase 3 or pivotal study is the one that is typically used to generate the definitive data that will go into a new drug application. So these are almost always larger placebo-controlled studies. The FDA technically requires 2 well-controlled studies, meaning they usually want the placebo in the study. In rare disease, they don't always require 2 pivotal studies. It's less common in rare disease than it is in common disease, but that is still technically what the requirements say. So finally, if you get through the phase 3 and you submit more data, you're on the track to an approval of a drug. So how does a drug sponsor get to market a new drug? So if they've run that gauntlet, they can get to the point of submitting an NDA or a new drug application. So this is the application that will allow them to start selling the drug. So this is a stack of papers. This is a very, very lengthy collection of all the data from all of the clinical studies, all of the preclinical data. This goes into understanding the benefit, the safety, how well and consistent the manufacturing is done, and ultimately contains information on the drug label, which is used to decide the label. So the label is what describes how and for whom the drug should be given. So if you've ever opened medication, you get that huge white fold out piece of paper. That's the drug label. And so all of this information helps determine what goes into the label. When a company or sponsor files an NDA, the FDA has 60 days to decide if they're going to even accept the filing. So I think they do a quick pass through it and determine if they see any major missing data or gaps. If they do, they can actually refuse to accept the filing. That's called a refusal to file. And they send it back to the applicant, usually with questions or requests for additional data. If it looks fairly complete, they will decide to review it, and they've got 6 to 10 months on average after that to review the NDA. And at that point, they can decide if they want to approve the NDA. Following a drug approval, the testing still isn't over. There's almost always a period of post-marketing surveillance where this will continue to monitor, especially for safety interactions, as the drug is delivered to a much wider group of participants than you have in a clinical trial. So, your standard clinical trial is really designed to be very homogeneous. So people as alike as possible so that you can control for all of these variables. When it goes out into the general population, you might expect that you could see side effects or other things that you hadn't seen in the clinical trial. So that will period of surveillance can last anywhere from several years to, in case of gene therapy, as much as 5 to 10 years after administration. There is, some people ask about full approval versus conditional approval. In the United States, full approval is the standard. Almost always when a drug is approved, is full approval. Under rare circumstances, you can get a conditional approval, which means that based on data from something like a biomarker, which we talked about earlier, the FDA could decide that it looks very likely that this drug is promising and could be approved, especially when there's a major unmet medical need involved. And then when the drug is approved, it's approved conditionally with the idea that the company will go back and continue to gather the data that it needs to show that there's a functional outcome that also shows this benefit. So an example in the US would be Oxandis 51, which is an exon skipping drug that was developed by Sarepta Therapeutics and by Dr. Mendel, who you will hear from shortly. That drug was actually approved conditionally in the United States because of the major unmet medical need and the promise that it showed. So there are many ways that the FDA can expedite this process. I'm not going to go through these in great detail, but you might hear these terms. Frequently, if a sponsor is awarded one of these designations, they will put out a press release. So you might hear about fast track, breakthrough therapy, or priority review. All of these things are designed to help a drug either give you more time with the FDA or decrease the turnaround time that it takes for the FDA to make decisions after various filings, or might give you more times to have meetings with the FDA. But they're all, for various reasons, designed to expedite approval. And it could be because it's an unmet medical need or because the early data look very promising. The last one, the RMAT designation, really does much of what the top 3 do, but it's specific for cell and gene therapies. But otherwise, it's very similar to those top 3 designations in the benefits that it provides. Other regulating bodies that you might come across if you're in a clinical trial, one is an institutional review board. These are made up typically of faculty members at research institutions, although there are private institutional review boards as well. But these are there to protect the rights of you and anyone participating in a research trial. So they want to really make sure that the risk of the study is justified by the potential benefit. All the local laws are followed, and they are also particularly involved in informed consent to make sure that if you participate in a trial that you really understand all of the risk and potential benefits and what you're getting into. The institutional review board has to approve the clinical protocol, that's the study plan, before that can go forward. So even if the FDA has approved an IND, the study still can't start typically until the institutional review boards. Sometimes at each and every site also approve that protocol. The Data Safety Monitoring Board is an independent group of experts, including physicians and statisticians, and often also patient advocates, who will review the safety and effectiveness of the trial as ongoing. One thing about the DSMB is even if it's a placebo-controlled study, they typically have access to unblinded data as well. The important thing is DSMB, based on what they find in their reporting, can stop a trial. So who all can stop a trial in progress? Certainly the FDA can. They can put a study on hold if they see safety concerns. The Data Safety Monitoring Board can recommend that a study go on hold as well when they review data. And an institutional review board can actually prevent a trial from starting. So these are all different checks and balances that are built in, mostly protect the safety of participants involved in the study. There's also another role that sometimes you'll hear about, an FDA advisory board or an ad board. These committees are committees that the FDA holds at their discretion sometimes if there are questions about a drug where the benefit risk ratio is maybe a little closer than they would normally, feel comfortable ruling on. So they want some outside advice. So these advisory boards are made up of external experts, usually with knowledge in the field, and they often include some people from the community who may or may not have the disease for which the drug is designed to treat. So again, another example of an ad board, there was one held for the approval of Exondys 51, which is the drug that I mentioned had a conditional approval. And so many people testified at that ad board. And ultimately, this is an advisory board. The FDA doesn't have to take their advice. They can listen to it, but in the long run, the FDA makes the final decision about whether a drug is approved. But it is one way in which the public can interact with the FDA and make their wishes known. Finally, access to drugs that are not approved. So there's a lot of interest in this topic. You might hear the terms compassionate use or expanded access. These are mechanisms by which you can have access to a drug that's not approved. Usually if there's some safety data available for it, often it will be as part of an extension of a study that's ongoing. That might be a phase 1 or 2 study, but the sponsor of the study can identify a population who's maybe not eligible to be in the main study, who could receive the drug through expanded access. There's also a less common pathway called an individual IND, in which a single investigator can submit a new drug or an investigational new drug application to treat maybe, I think it's up to 3 to 5 patients. If there's a major unmet medical need, there's also, you can do this on an emergency basis. So these are some ways in which the FDA is a little bit more flexible sometimes with access to unapproved drugs, but you really have to justify it well. And the important thing to know is that the drug sponsor ultimately has the right to say yay or nay about whether the drug can be made available for this purpose. So it's not the FDA who controls this. You may have heard about the right to try laws. Many of the states in the United States, I think over 40, have the individual right to try laws. And now there's a federal law that also says you have the right to try drugs that are not approved under certain circumstances. This is the same situation as the actual existing FDA regulation, in that the drug sponsor has to agree to make the drug available for this purpose. So ultimately, you do have the right to request it under either mechanism, but the drug sponsor has to agree. So this is just to summarize again and say, so this is the process we've just talked through. Again, it's highly regulated. You can see the different places where the sponsors have to submit data. There are a lot of ways that protect your safety as a trial participant, and it's quite a gauntlet, but it does, in the long run, the whole system is designed to save us from incidents like the elixir that killed 106 people. You really want good evidence that a drug is safe as well as effective. So with that, I will say thank you. And there are a couple of additional resources listed here, both of which are really good basic primers about clinical trials and FDA fundamentals. So thank you very much. And I will turn it back over to Michelle now.
Thank you, Sharon. I would now like to move to our next speaker, Dr. Mendel.
Sharon, I enjoyed your talk a great deal, and in many ways, our discussions here will overlap. You have a more technical description, and perhaps I hope I can complement what you said with a practical application. And in that sense, I'm going to talk about how to get really from the bench to the bedside. And I think one of the things that's underappreciated is how planning starts at conceptualization before any lab projects start. If you're going to think about doing, I'll use gene therapy as the major example. I've been involved in translational research for many years, but over the last 20 years, I've focused on gene therapy. And in that context, there are 3 conditions that should be met when you're thinking about doing a clinical trial. And this is how I generally teach people who are coming through our lab for direction in moving their career forward. First of all, gene therapy should be directed at a disease where there is no known treatment. It's a significant step to do gene therapy, and there are certain downsides that we'll talk about, but you really need, you're talking about risk-benefit ratio, and in that context, it should be something that has no current treatment. Secondly, you need an animal model that closely mimics the human disease. Often this is a very high bar to set, and we sometimes have to compromise, as we do necessarily for Duchenne muscular dystrophy, as I'll mention. And then you want to have a plan in mind that's feasible. Is the outcome likely to be favorable? In that context, I like to put it in the phrase of cost-benefit ratio. That means that there's enough patients to treat. You don't want to direct a gene therapy trial at a very small number of patients because it's very expensive. And so you want a significant incidence of disease. This is not to diminish the importance of rare diseases, but you need, these trials are very expensive. So in order to get partners with industry, you want to have a significant number of patients who are affected. And I like to put it in the context of a very famous sporting event, the Masters 2005, at which planning should predict a favorable outcome, just as Tigers pitch on the 16th green at the Masters tournament. So keep these things in mind. When you're planning, you want something that is fairly secure in what you think will develop. I'm going to talk about this specifically in relation to Duchenne muscular dystrophy. It's a disease that I'm highly passionate about, and I have committed myself to stay in this field in the gene therapy world until we really have significant outcomes and make a difference for boys with this disease. It's first of all the most common form of childhood muscular dystrophy, so it meets expectations about cost ratio benefit, and it fulfills as criteria, as I mentioned, for cost benefit. And it's an example to show how the gene therapy is done, physically done, and I'll walk you through those steps. And it's important because not only is this relevant for Duchenne muscular dystrophy, but it's other muscle diseases can be treated in exactly the same way with different genes and perhaps different viruses that we'll talk about. But the methods are applicable to other forms of muscular dystrophy. And in fact, we're heavily invested in multiple gene therapy trials. The SMA trial was summarized briefly before, and now we have the Duchenne trial, and we're also involved in limb-girdle muscular dystrophy trials. So further consideration in planning is, if the gene therapy is proposed as a treatment for disease, what are the usual questions we get asked? I like to put this up front because this is what patients think about and what we have to think about when we start a gene therapy trial. Where do we get the gene for testing and treatment? Very common question. How does it get to the muscle if this is a Duchenne muscular dystrophy trial or a limb-girdle muscular dystrophy trial? Or there's another target, how does it get to the target? And ultimately, is gene therapy safe? And Sharon emphasized this in obtaining an IND, and it's absolutely critical. And then another question we get for gene therapy is, how many times does it have to be given? Gene therapy is different than Exondys 51, which can be given weekly. It's different from prednisone, which is given daily or for some patients weekly. But gene therapy is unique. It's a one-time treatment. And so that needs to be emphasized, a very common question that we get asked. So let's look at the history of gene therapy. Prior to 1999, when I did my first gene therapy trial, it goes way back 20 years. And the main approaches to doing gene therapy were, and as I'll explain, we put the gene in a virus, and the virus then carries the gene to our target tissue. the main viruses that were used were retrovirus and adenovirus. And the reason these two genes were liked for delivering or for delivering the gene was that the size could accommodate most genes. And that's a huge advantage. But the limitations were illustrated by historical experiences. With retroviruses, there's something called insertional mutagenesis. And what that means is that retroviruses, like HIV, insert into the human genome. When they insert into the human genome, this is insert into other chromosomes, they have a very secure place, but they might disrupt another gene. And in one of the first clinical trials done with retrovirus for severe combined immunodeficiency disease, several of the patients, in fact, 4 of the patients ended up having leukemia and 1 died. And that tended to push away retrovirus as a vehicle for delivery for what we call monogenic illnesses. It's still used for treating some cancers, but it's not used generally for genetic diseases that are caused by a known gene. The other problem is with adenovirus, and this is an important distinction by name. Adenovirus is a gene, is a virus that's very large, and it can carry the whole DMD gene, Duchenne muscular dystrophy gene, which is the largest gene in the human genome. But it's ideal for that. But nevertheless, one of the patients who was treated in 1999 with adenovirus delivering a gene called ornithine transcarbamylase, the patient developed an exaggerated and very significant immune response and died on the 4th day after delivery. And that's a famous case. Using his name here is not without respect. It's one that you can look up on any internet, and it'll describe the Jesse Gelsinger case. So that tended to push the field away from retroviruses and adenovirus and ushered in the adeno-associated virus as the gene delivery. Let me emphasize that adeno-associated and adenovirus are not the same. In some ways, it's unfortunate that they have similar names because people get confused about this, and the obvious reason is there. But there is a reason for that, and that is that adeno-associated virus requires a helper virus to express, and adenovirus is one of the helper viruses that's used to help expression, and so it's adeno with an association, and that's called AAV. The problem with adeno-associated virus or AAV, as I have shown on the descriptive picture below, is it has a very small packaging capacity. So if the human DMD gene is 2 million base pairs or more, and when its messenger RNA cDNA is 14,000 base pairs, it can't fit in a virus that will accommodate only 5,000 base pairs. So that gives us a problem right from the outset. So if we look at, but adeno-associated virus has now become the gene of choice for basically all human monoclonal or all human monogenic diseases. And the first step in order to make the virus work for gene delivery is that the adenovirus is also adeno-associated virus, and let me re-emphasize that if I mess this discussion up because I use these terms overlapping, please forgive me. AAV is what we'll call this. AAV has only 2 of its own genes, and those are called rep and cap. And the reason they're called rep and cap is when the AAV is delivered with its own genes, rep and cap make a new capsid for the virus or a new capsule envelope for the virus, and it allows the virus to replicate. Human gene therapy is not a usual infection. it removes the viral genes and puts in the gene that we want. So it's removing rep and cap. So the virus is essentially naked. It only has the shell of the virus. And what we have to do is load the shell of the virus with the gene that we want to use. And that's called the transgene or the cassette. It goes under a number of different names, but I have a picture of what this looks like. If you look over at the picture that goes from above that from the N to the CT, that's what the normal gene looks like. And the gene that we put in is small and has only, see if this will show up, yeah, has only this part here, the ABD, that's the amino terminal, analogous to the N terminal you see here. It has hinges, 1, 2, 1, 2, I'm sorry, 1, 2, and 4. That's shown here, 1, 2, and 4, and 1, 2, and 4 here. But notice that almost the entire middle of the gene is gone now. And it has only these repeating units, 1, 2, and 3, and 24. So it has 4 repeating units and 3 hinges, and that makes up the gene. Now, how do we know it works? In the development of this process, it's been tested multiple times. In our own labs, we've tested it probably at least 1,000 times, and we know that it works to significantly reverse the disease. But animals or mice, as we usually use it as the target system, are not men, as we say, in a very distinct way. Mice are not men, and so we can only do a certain amount of prediction from preclinical trials. Now, once you have the gene identified like this, and you have the virus that you're going to deliver the gene with, and I should mention one other thing. I'm not sure I have it in another place, but the virus that's devoid of its own genes and the gene that we are going to put in called the transgene, those two things together make what we call a vector. So the vector is the capsid of the virus, the outer shell of the virus, plus the gene of interest, and none of the genes that the virus would normally carry. And we take that to our vector manufacturing facility. Now I have a picture of this here because this is a critical step in the development of gene therapy. And I've been at Children's now for 16 years. Ohio State and Children's Hospital are linked, but I came to Children's full time 16 years ago. And when I came, I had already done my first gene therapy trial when I was, what we say, on campus, and then was asked to develop the program full time at Children's. And that appealed to me because my targets were, as we now know, SMA and DMD, both childhood diseases. But I went to the CEO of Children's Hospital and I said, we aren't going to be in the gene therapy world unless we can make our own virus. This is a very expensive, detailed process. And we put together a vector manufacturing facility, and Sharon referred to this as a GMP. This is really what we referred to in the field as a GMP. And so we have these various units here in the GMP that can make the viral product and make sure that it has rid of all of its impurities, that it meets all requirements of the FDA, and anything coming out of this unit has to be meet FDA criteria. And then I want to show this picture here too to give you some perspective. We have all of these serotypes of virus. Now, a serotype means that the capsid of the virus, the envelope of the virus, has different amino acid structures. And we can type those. That's where they get these numbers. Here's AAV1, there's AAV2, and so forth. And now there are 100 different serotypes. And these 100 different serotypes can target different tissues. These are the common tissue common AAV serotypes in the targets that I'm showing you here. But here's skeletal muscle, which is what the subject is today. And all these serotypes will be able to express and target in muscle. But here's AAV RH74, and that's what we use. This is a bit of a distinctive serotype, RH74. It is isolated from non-human primates, that's where it gets its name RH. And the AAV 74 is a number nomenclature. And the reason we use it here at Children's Hospital is because it was isolated here at Children's Hospital. Many of these other serotypes were isolated by other investigators all around the world. The most number was by Guangping Gao when he was working with Jim Wilson. And there's in the field, for better or worse, every time you isolate a serotype, you patent it. When you patent it, you essentially own it. So if I want to use an AAV9 serotype, as we did for SMA, I've got to pay a patent fee to the person who patented it. This one here, we don't have to do that with because we patented it internally at Children's Hospital. So it makes it more easier to use in the long run for us without having to pay an additional fee for its use. We also know from vast experience now that it targets muscle beautifully, whether we inject it IM or through the circulation. We get very high levels of gene expression, as you'll see. So Sharon also mentioned this. Before you can do anything, you have to show that your vector, as we call it, in this case, here's the AAV transgene. That was the gene that we put in. Remember, when we inject it, this gene now is going into this virus and the virus now has none of its own genes. It has only the gene that we put in it. And we make this in the GMP in millions of copies, put it in a syringe, and the first thing it takes we go back to the lab. And when we go back to the lab and inject it into muscle, either directly or through the circulation, follow those steps again, we inject it into the muscle, and it courses through the muscle fiber into the nucleus. The capsid then is emptied, and the capsid doesn't enter the nucleus, it's degraded, and only the DNA is. The first step in that process is often that the DNA, it depends on how you deliver it, but in most cases the DNA is delivered in a single-stranded DNA, and then it has to be made into a double-stranded DNA in the nucleus, and this double-stranded DNA forms an episome, a circular form of DNA. And essentially, this is separate now from the chromosomes where HIV and these other retroviruses would insert. So it's safe in that sense. It has no ability to disrupt other genes. And in the double-stranded DNA, then, it then forms the protein of interest. And in this case, this is called now, since it's not the full-length dystrophin, it's called micro. And this is, so the gene now in this double-stranded DNA forms microdystrophin. And now when we inject it into the mouse, what we hope is that this mouse now has outcome measures that we can measure and are better. The mouse is a reasonable, not an ideal model for muscular dystrophy. That's why in many cases, you have to look carefully at the outcome measures that were used for mouse studies. But we also like to see that this same process took place in canine dystrophy. And it was also done in primates, so that the more assured you are that you have multiple outcomes, the more likely you are to be successful in your trial. Once this is done, we have data that shows that experimentally our gene is working, and we then have to do what we call a formal toxicology studies. Formal toxicology studies means that we take 10 times the amount of virus that prove to be effective in our animal studies and show that it's safe in the various species. I have a list of pictures here of how many times we use this in order to prove safety. And we've done it. This shows an example. This is in primates now. This has been done 142 times in our laboratories here at Children's Hospital. And looking at all the organs, not only the muscle in this case, but looking at heart, diaphragm, liver, spleen, lung, gonads, kidneys, lymph nodes, to make sure that there's no pathology that we can see. And then we take all this information, the experimental data, the planned data, the experimental data, the clinical trial design, and all of these things are put into the IND that Sharon mentioned. And this is an actual picture of an IND. I like to show it because it shows how much work goes into it. This, in this case, is an 8-inch document, and it has to be almost hand delivered to the FDA in order for it to arrive. Now, it'd be heartbreaking, as Sharon said, for this to get to the FDA, and the FDA says, sorry guys, let's try again. You need to do more experiments or this or that. And it happens. But in any case, this is what it looks like when it goes to the FDA. And then if it's approved by the FDA, then we take it to our IRB. And I don't need to reiterate much of this. It's the security safety board for our patients, and considers ethical considerations. Ethical considerations are sometimes pretty, I'd say pretty meaningful or pretty hefty, in the sense of, is this something that's likely to work? I mean, the IRB is going to think about that. They're going to think about whether this is really a practical solution to the disease, and they have the right to reject this. They also have the right to reject, if you're thinking about an IRB for a product that's been shown to be effective, do you need to do then a placebo-controlled trial? These are all considerations that go into the IRB approval. And we also have another local institutional biosafety committee that employs NIH guidelines to provide oversight for any research using recombinant products. Just to introduce you to the term recombinant, means that the virus that we use is recombined. So now it's recombined with the gene, and now it becomes a recombinant product. So let's look at the next step. The next step is to carry it. The IRB then actually has approved this product. The FDA has approved this product. And now the patients have to approve the product, and the parents and patient have to consent for the clinical trial. The IRB approves the consenting process and the consent form as we use it, to make sure that patients understand gene therapy, and then if they understand gene therapy and sign the consent, then they have to pass eligibility requirements that are part of the protocol. They have to be sure they have no antibody to AAV, and normal blood tests and a normal physical exam. So let's briefly look at these other two important steps. Antibody has gotten to be a major, in some ways, a major challenge and even a barrier. A certain percentage of patients, if we use an AAV, and let's say AAV RH74 or AAV9, which have been two successful ones now, one in SMA and one in DMD, if we look at these serotypes, they're different, and patients can have pre-exposure to these AAV, to these virus serotypes. It's important to realize, though, that this AAV does not cause a human disease. It's a silent infection, a little bit like we've heard about COVID-19 can be a silent infection. But AAV, as opposed to COVID, doesn't have its own genes, so it doesn't cause any. It does for gene delivery. It doesn't cause any disease, and even in its wild type, it doesn't cause a human disease. But the exposure can be detected by pre-existing antibody, and we must check for that. And if there's pre-existing antibody, here's a picture of an IgG molecule that's localized, that localizes to the AAV, and then it prevents delivery then of the virus. And this must be done for any serotype that you're going to use. And if it's an antibody that's to one serotype, it doesn't mean it's an antibody to all serotypes. That's an important concept. So it must be done for any clinical gene therapy trial that's to be done. And the antibody test requires a blood test, and it takes about 4 to 7 days to get the lab results back. And I also mentioned about that there has to be a check for screening for any disease process. This shows the major ones we look at are, is there any kidney disease? Is there diabetes? Is there any liver disease? And those are probably the predominant ones. And we can use, muscle is challenging because AST and ALT are two enzymes that are shared with muscle, but we have some opportunity to look at muscle, look at liver independently. I said that wrong. Liver enzymes are shared with muscle, and we have to be sure there are no liver complications. So we have the opportunity to look at liver alone with GGT and GLDH. And if you're involved in a clinical trial in any way with muscle, you'll see that these are very important outcomes. So you want your patient not to have any GGT elevation and not to have any GLDH elevation. They will have ALT and AST elevation as part of their muscle disease. I hope that's clear. Then we do a muscle biopsy pre-treatment. This is an example of how the muscle biopsy is done. In our center, we always put the patients to sleep. We mark clearly which muscle. The common ones we use are the calf muscle, and we use the anterior muscle on the lower part of the leg, the tibialis anterior. We use the quadriceps muscle, and we use the biceps muscle, depending on the trial. And this shows that we do this, when we do the biopsy, we monitor with ultrasound. This is the needle biopsy that we use. We prefer needle biopsies because it takes a decreased size of the sample. By having a decreased size of the sample, then we are able to spare muscle, and we get enough of a sample that we can then test for the amount of gene that we delivered with microdystrophin expression, either by direct staining for counting the number of fibers present, or Western blot to quantify the amount of dystrophin that's present. It's a very valuable approach to use a Pecora needle biopsy. And then this was actually the first gene therapy trial in DMD that I did back in 2006. I had done actually a limb girdle trial actually before this. So this wasn't the first trial I had done, but it shows by example. This was a direct intramuscular evaluation monitored by ultrasound with an interventional radiologist standing by to help isolate the muscle. We're no longer doing these direct intramuscular injections. For the most part, we do vascular delivery. It's not unusual for a gene therapy program to start with a safety trial first in muscle, then a limited vascular trial, then a systemic vascular trial. And this is an example now of a systemic vascular trial, which we do routinely now. This shows the virus as it's delivered from the pharmacy. This is the actual volume for this patient that's calculated based on weight. We load this into an infusion pump, like you see here, and then the infusion pump then is set for how much we want to deliver. In general, we deliver the virus over an hour to an hour and a half. The patient is awake during the delivery. There's almost no feeling from the delivery as we put the virus in. And then here's exactly how it looked. This is a real picture of a real patient getting a virus. It's delivered in one arm. We keep an IV open in the other arm in case there's any blockage or anything that should occur here on delivery, and we can switch this over very rapidly. We monitor heart rate and basically an EKG and respiratory rate and temperature all during the trial. And then after the test is over, we monitor blood tests, the same ones I mentioned. We monitor kidney function, glucose function, monitor liver enzymes. Those are the ones we concentrate on most. The ALT and the ALT are elevated as part of muscle, but at the baseline, their GGT and their GLDH are normal. GGT here, GLDH here. Elevated AST and ALT from their muscle, normal liver function. Post-delivery, though, we commonly see liver enzyme elevation. And I won't discuss all about that. That can be part of a question period. But liver enzymes are a common form of AAV, what we consider an AAV adverse event. We suppress these elevated liver enzymes with prednisone, even though the patients on Duchenne are on prednisone. We'll give additional prednisone to block the liver enzyme elevations. It's a manifestation of inflammation. They feel nothing from it, incidentally. There's virtually no systemic manifestation of this in terms of patient toxicity or patient symptoms. Now, I like to show this picture because this is an actual picture again of a patient who we just finished delivery on. I like taking the picture here. Shows the patient has virtually no feeling, had no problems during delivery, is happy as a lark, so to speak, and not any problem. But shortly after gene delivery, the common side effects are we do see nausea and vomiting, usually starts around the second or third day, may persist up to 3 or 4 weeks, And we often have loss of appetite, elevated liver enzymes. We may have transient platelet decrease. All this is, the liver enzymes and the platelet decrease are not symptomatic, but of course nausea and vomiting are. And then during the patient stays in the hospital usually 1 day after gene delivery. And then both pre and post gene delivery, we will do outcome measures. The most common one we do that we rely on is called the North Star Ambulatory Assessment. These are 3 of the tests that are part of the North Star Ambulatory. This patient here is scored in how well he stands up. This one in terms of lifting head and trunk control, and this one stepping up on a step board, as you can see here. In addition to these, we do, and Sharon mentioned this, other outcome measures are the 100-meter walk run test, climbing stairs. These are time tests, very quantitative, and allow us to, between the North Star and these other measurements, we can, comparing pre and post gene delivery, measure efficacy. And this is just quickly show you, this has been published now, so it's not embargoed any further. This is our 1-year clinical trial in the recent systemic gene delivery. All measures have improved: the North Star, time to rise, 4 standard stairs, and the 100-meter. And we're now into a double-blind randomized controlled trial that has extended this now to additional 40 patients. And show this is from our current trial, and I like to show it because this is 18 months post-gene therapy, and we can see how well he gets up from the floor. And finally, this is the gene expression data from our first trial. We have very, very high levels of gene expression in the muscle that reached 80% across the board, as you can see here. And this one actually reached over 90%. So a very favorable outcome measure here. And so this is how the clinical trials work in progress. The collaboration is between patients, families, and all of the staff in the Gene Therapy Center. These are the prominent players in our Gene Therapy Center who are involved in the clinical trials. And also this trial has been done with Sarepta as the industrial sponsor, and two people to highlight there have been very helpful. Louise Rodino-Claypack, who's the scientific director at Sarepta, and she was one of my former fellows and now has far exceeded me in terms of an industry placed position, and Teji Singh, who's one of the leaders in the clinical trials at Sarepta. So these are the experience, and I'll end there and turn it back over to Michelle.
Thank you, Sharon and Dr. Mendel. We sincerely appreciate your time and expertise and everything you do for the neuromuscular community. The information you shared today was incredibly valuable and education. Also, thank you again to our event sponsors, Genentech and Mitsubishi Tanabe Pharma America. We would not be able to provide events like this if not for your generous support, so thank you very much. If you have any questions around the MDA Engage program and/or items from this webinar, please feel free to email them to mdaengage@mdausa.org, and we will do our best to answer them. Do also remember MDA's Resource Center is a wonderful resource for MDA's families. The MDA Resource Center is available to provide one-on-one support via phone or email for individuals and families looking for information about the diseases in MDA's programs and more. This concludes the MDA Engage webinar, Clinical Trials from Design to FDA Consideration. Thank you very much for watching today, and we look forward to you joining us for future webinars and additional educational events.